conversations that we had, we really were talking more about context, and we were talking about the different ways that context, primarily neighborhood context and school context, measure like different measurement strategies, and we were also talking in that about how Candace has been one of the innovators of trying to think of new ways of measuring context. So besides looking at using surveys or federal data to measure school composition or things like that. And the way that this is moving is using different technologies to track context that's more individualized. So for example, you and I might live in the same neighborhood, but we, on a daily basis, are in very different parts of that neighborhood. And so using technologies to track what actual my daily context is and then measure things about that. And also thinking about more about how the virtual context has changed our interactions with concrete context. And one of the things about this is that as we move to that much more fine-tuned sense of context, school context or whatever, or neighborhood, the chances that those things are influenced by selection, by genetics, selection into the context grows because they're more individualized. And so that led us to a discussion about this connection, about how people, especially in social science, when they get into this gene stuff, what they're really, for whatever reason, they're interested in gene-environment interactions. But we talked more about genetic selection into context and how those two things go together over time, continuously in a feedback loop. So we're going to talk about that. Or they're going to show some slides from research that's relevant to those issues.
Okay. So yeah, thank you for setting that. So we're going to end on that, on the context, but we're going to go through, we're going to start a little bit actually where you guys left off. But I'm going to put up a slide that anyone who's seen me give a talk ever has already seen. So the biomarker hype curve, you've seen it before, you've never seen it, Paige, you've probably seen it, Elliot. So I work in this world of integrating all sorts of biomarkers, including genetics, into social science studies. And I can just tell you that there's, it's both as a new biomarker gets created and also as a biomarker that may be old in one field moves into a new field. There is this hype that gets connected to it. Whereas you see a new innovation in the field and it over time, it really quickly gets a lot of visibility and everyone kind of thinks it's going to solve all of our problems. Then we start to figure out what the problems are with that measure and people get disappointed or start to throw it out. And so I have all sorts of examples. Actually, every single thing that we've talked about, someone mentioned epigenetics. That's definitely going through this right now. GWAS, when it first started, definitely went really high up, a lot of expectations, and then it dropped down into this trough of dissolution that now it's probably found. I would say we're definitely in the plateau of productivity. But when we move into a new area, it kind of goes through this again. So part of it is just to keep this in mind as we go through this. I will say this group has been far better than almost any group I've heard in talking about this and being aware of some of the issues before they get going into it. So just keep that in mind. So the other thing is when we start talking about context, and again, one of the things that I try to work on are these larger population-based studies and helping them to collect is whose data do we actually have? And by that I mean whose genetic data do we have, but really whose biomarker data. Just to be aware, most of this biological data work is not done on population-based studies. The vast majority is actually done on either clinical studies or community-based studies. There are some groups like SSGAC, which has a lot more population-based studies involved in it than others, but just to be aware, not all data sets are what most social scientists would consider population-based. And there's lots of research on this. It's also the difference between recruiting versus sampling. There's a lot of the studies are recruitment-based. So they put out flyers and just try to get people to participate. So you end up having a very unique distribution and sometimes people call them weird samples, Western, educated, industrialized, rich, democratic. So there's, again, lots of citations just to keep this in mind. Let me go directly to genetics though. Why it might matter, I think you guys can understand that. So this is Martin et al. So this is on bioRxiv. It's something that we've known before, and there are a few other papers that are like this. I like this figure. It's a more recent figure. So I'm not involved in this work, but it's pretty self-explanatory. You have the x-axis is just year. Y-axis on the top graph is individuals in GWAS, but there's a lot of double counting of individuals, right? So this is just a count. And then you have by different populations, European. Otherwise, those solid bars on the side are what the distribution is in the world. So it's a very clear, as we've been talking about this European ancestry, very different distribution of where that work is being done. The bottom graph, by the way, is a fraction instead of just a count, because you do have double counting. So I think you can see pretty clearly what the story is, where our data are coming from. So keep that in mind. That's whose data we have. Why might that be a problem? Well, the reason why I think this is important as we get directly into context is genetics and location are very, very closely connected. This is a paper that I can't believe it's now a decade old, but it's a foundational one where I think it was 3 generations. They had to have families living in Europe for 3 generations. They could then pinpoint like where they were based on their genotype, and it's pretty good at mapping Europe. So just keep that in mind when we start talking about context, that our genotypes are connected to our context. And that makes perfect sense if you're a demographer. For most of human history, the person that you mated with was probably living pretty close to where you are right now. So these things make sense from a very formal demographic point of view. But that's important in considering what context means when we start doing genetic studies.
Coulter, maybe just to clarify, because
Sure.
people have varying levels of familiarity.
That's true.
Right, so these are not scores on the genetic propensity
Yes.
for a particular outcome. These are actually kind of just the frequency of DNA variants that
Yeah.
may have nothing to do with any outcome that we're interested
Right.
in, which just tend to be overrepresented in some demographic groups relative to others.
Right. No, so I mean, literally, this is not meaningful in the sense of predicting anything other than place, really. And so that's just key when we start thinking about gene-environment interactions. This is genetics and context are linked for the main reason that they're just physically linked together. So what does that mean? What that means is, and there's another paper by Martin, but we've done this in the Fragile Family Study too. So the Fragile Family Study, I think I've mentioned it a couple times. It's a birth cohort study in the U.S. cities. It's representative of U.S. cities, kids born 1998 to 2000. We've been following them until now. So the last one just turned 18, which was nice in September. Anyway, we have polygenic scores for them. It's a very race, ethnic, and SES diverse sample, which is unique among these. So this is a polygenic score for height, and again, like I said, Martin has a nice set of papers on this too. So this is the phenotypic. This is the height. It's normalized, but this is by different racial groups. We could do it by ancestry groups too. Race and ancestry are correlated, but not the same thing. We can talk a lot more about that too if we want, but that's height. And so really, in fragile families at age 15, kids all have these groups have similar distributions for height. However, if you look at the polygenic score for height, because of these differences in allele frequencies related to just, not necessarily related to height, obviously, but related to geography, you end up getting very different distributions for the height polygenic score. So much so that, so you have European ancestries would look very tall, and African ancestry would be much shorter than them, and you'd have some other groups in between. Now, we know that that can't be true, and this is why we don't compare across ancestries these scores, because the means would be very different. Happy to give Pietro.
How much is it in centimeters? Like predicted centimeters, the difference between.
So I think for adult height, I can't remember what it is in the Martin paper, for fragile families, it would be about 4.5 inches at age 15.
4.5 inches. Okay.
So there'd be 4.5 inches from the two means, which is
It's
clearly not true, but you start to see that difference.
Yeah.
And it doesn't mean that it's not predictive within group, and I'll show you that in a second, but you can't compare across groups for very clear reasons, but it doesn't necessarily mean it doesn't lead to some problems. Just to also give you a sense, when we say African ancestry, there's actually a lot of genetic diversity within these different groups anyway. Don't worry about the color coding as much. This is based on kind of ideal HapMap 3. So these are populations that are supposed to be very different genetically. And so if you actually start with it looks like it got shifted over a little bit. The far left in fragile families is the white sample. Self-reports as white, and really those two different colors are either they're Northern European or they're Southern European. And that's pretty much the only two groups that are there. But you go to self-reported black, there's a mix of African ancestry and a fair mix of European ancestry in there as well. And then if you go to our Hispanic sample, which is kind of in between those two, very, very diverse. A lot of mixes of different European, African, Asian ancestries. So keep all of that in mind when we go to, this is just to kind of piggyback on some of the other things that people were talking about. Okay, so I just said do not compare the means across these scores. This is not comparing the means. This is the kind of the effect size in these different populations. So this is the educational attainment, most recent one, polygenic score, by different ancestries. So EA is European ancestry, African ancestry, Hispanic ancestry, and then because we have such a diverse sample, we actually, our admixed population, which means it's you can't classify them into one of these other populations, is actually larger than our European ancestry population. So because of our sample. So it's kind of, we have these four different groups, and this is how much that polygenic score explains of different math-related tests that we have in fragile families. Yeah.
So is Hispanic, is that defined as people who say they're Hispanic, or those are people who have predominantly American continental ancestry?
Yes, it's the second. This is based on their, I mean, it overlaps pretty well, but it's the genetic ancestry, not the self-reported. Yes.
How is the model for the polygenic score trained? Is it trained based on the European ancestry?
Yes,
data?
yes.
So this is basically just because of the overfitting to the.
Well, we could go into that a lot more, and Patrick, if you want to chime in on this one, I mean, so there's a couple different reasons why this could be, right? So it's trained on the European sample. The reality is that the chips that were designed, especially for these older cohorts, were particularly attuned to European variants.
What's a chip?
A chip, the genotyping chip. It's the actual way that we take the saliva and turn it into.
The chip determines what's actually, like, which SNPs are measured.
Right, so you have, you know, your genome is a series of DNA letters. We're not actually measuring every DNA letter in your genome. You're selecting which DNA letters at which locations to measure. Which variants, which differences between people and DNA letters are present in people of different continental ancestry groups varies. So you might have variations that would not be present in anyone from Africa, and someone from Africa might have variants that are not present in you. So when you're designing a chip that picks which DNA letters to measure, you're privileging ones that we're going to find in a European population.
So you're using what's information that you already have to decide which subset you sample, but it's information about a certain population.
Yeah, so out of about 2 billion DNA letters, we think that like maybe 15 million are variable in 1 out of 100 Europeans. And they range in their frequency from, let's say, 1% to 50%.
Because it'd be a waste to look at ones that don't vary at all.
So the genotyping chips then
Correct.
select from among those 15 million common variants between, the earlier chips were like 50,000, but now they're, routinely a couple of million variants are measured, and those are selected, but what you're saying essentially is based on frequencies in Europeans, although now they sell GWAS chips that are.
No, they're better, but the
Yeah.
problem, so the thing to keep in mind though is that most of our GWAS results are based on chips, at least earlier, that privilege these other measures. Now, they've gotten better, and the imputation has gotten better over time. Imputation is the way in which we take what we've measured and impute those using correlation structures in the genome, impute nearby SNPs that aren't measured.
It is worth noting that, when geneticists say imputation,
It's different than,
they're often, they mean a process that has more precision often than when, as social scientists, we're imputing unmeasured data.
these are things that are correlated at 0.99, 0.98, 0.95.
Not 100% even a lot of
Yeah.
locations. You can know with certainty.
Yeah.
So when you say something like code the entire sequence, what do you mean?
So chips are taking a selection of your genome. Sequencing is literally trying to measure every single one of those SNPs. Every single
So
one of
it's
those
billions?
experiments.
SNPs.
Yeah,
So
so
the billions.
Yes, every single, yeah,
Every base pair.
every single base pair.
So all
Not
2
just
billion
SNPs.
letters.
Yes, all 2 billion.
Like your DNA sequence is 2 billion letters, of which a small fraction, let's say 15 million, are commonly variable between people, of which we measure a sample of, let's say, 2 million. But having measured those 2 million, we can generally infer the values of the other 13 million with a high degree of certainty.
Right, so sequencing is really trying to get that really fine grained.
And in fact, to get a really good quality measure of 2 billion DNA letters, we have to sequence you not once, but about 40 times.
Yes, yeah. If you want to do a clinical level, it's 30
So we have to generate,
So just to clarify one comment from Taipei, so the polygenic score was trained on Europeans, but not on these Europeans,
Not on
on
the-
a meta-analysis.
Right.
So those R-squared values, even for Europeans, are not due to overfitting.
Right.
To this day,
Right,
sample.
right, so
That's
these days, I mean, so that's the other thing to keep, so these are kids. There weren't any kids, I mean, what was you guys didn't pick anyone over or under age,
30, I think.
30.
They had to have finished their.
didn't have to finish school, right? So so they couldn't have been in there even if they had existed before, anyway. So yes, thank you for that. So anyway, this is just the partial R squared or the incremental R squared of just that polygenic score.
There's a lot of other reasons that they're different. I don't
Yeah.
know that we need to go into all of them, but there's a lot of technical, and then also, you think that G by E is a thing. It's a bit, I mean, like, it's,
Well,
if you think it's an important
just
thing.
a scale
here.
transformation, right?
So,
Just a picture argument.
But I mean, like, I think it's second order to all the other things is what I mean. Like the technical issues of correlation structure differing and all those things I think are more important to describe these differences than just that there are differences in genetic types.
So yeah, and I wanna, did you wanna say something else?
I just have a question.
Okay.
Would the proper interpretation of this graph be then that for African Americans and mixed, that the predictive power declines over time, and likely that the context then comes into play more over the life course?
So I think there's a lot of, I mean, this is brand new stuff that we're trying to tease out why it's different. I mean, so in comparing over time, African, so first of all, let me just explain. So this is the age 5 Woodcock-Johnson applied, is it applied math? Or applied math problems at age 5 and at age 9. In age 15, we didn't have that. We had their math grade, the reported math
So
grade.
that could be just a.
Yeah, so it's not necessarily always comparable.
But there still looks like a decline between
Yes.
5 and 9 for African Americans.
Yeah, there is a significant decline there. Why that is, what causes that, we haven't really sorted out yet. This is new stuff, preliminary stuff that we're just sharing with you guys.
I mean, so like this could be totally non-defensible speculation, but and it's like this could be an artifact of something about how the thing was created. But if it in the future, might someone be able to look at this and say, whatever it is that's predicting success for whites in America is, suppressed in some way because of discrimination or disadvantage for African Americans. And so the kinds of thing like the kind of Protestant work ethic, work hard, get ahead, is more true for some groups than other groups given stratification.
Or the African Americans are less in a position to fulfill their potential.
So this relates to what Patrick was saying,
Yeah.
which is that's where we would all love to, those are the hypotheses we love to test, but there are technical reasons that we can't necessarily draw that conclusion from this. If we had trained the study on Asians, East Asians, we would see the same figure, more or less, where the highest bar was for the Asian American. And the question is, once we account for that, which right now we don't have a way for accounting for, then the question is, would we still see this sort of differential prediction, in which case your explanations would be very, very plausible.
Right.
And we're not ruling them out, it's just that we can't
We
distinguish.
can't rule out these other.
We have other lines of evidence that are compelling that, the structure of opportunity is different for blacks and whites in America. We honestly don't really need genetics to tell us that. And so maybe toward the end we can talk about how plausible it is that they'll ever be able to tell us something. But right now, we can't make any interpretation of that. Unless you're Jason.
Right. So there are some people who would say like, oh, this is because of these social things that you guys were just describing. Yeah. But my point was more to say, just because we say they're not as predictive doesn't mean they're not at all predictive. So they are predictive of these kids, even though they weren't trained in that sample, even though all of these other things, even the technical problems, they still are predictive.
And your lowest R-squareds are higher than the R-squareds that we studied, when we were looking at the first iteration of the educational attainment polygenic score.
Yes. Yeah.
And so in some sense, this could, even if you just run 2 separate regression of G by E, you could still have the G being this polygenic score and say there's a reasonable heterogeneity within the African American sample that can be picked up by this.
Yeah.
I cannot compare the 2 coefficients
Can't compare
approach,
the two,
but
right.
Or
That means-
you could potentially study if you thought that there were more and less discriminatory educational environments that black kids are growing up in different fragile family settings. You could ask whether the apparent penetrance of these genetics is higher in the low discrimination schools than in the high discrimination schools.
Right.
And you can ask these questions in twin studies about these problems.
Yes. So anyway, I just wanted to make sure, so again, this ties us, as we start thinking very specifically about which interventions and which contexts, this is a key issue. So we kind of already talked a lot about these things, gene-environment correlations. Before you even get to gene-environment interactions, I think Dan has actually really did a nice job of, after we had a whole bunch of papers, and I'll show you a few reviews of all of these gene-environment interactions, really pressing forward and saying maybe gene-environment correlation is the first place to start, and so there's some really interesting things there. We talked about active, evocative, and passive, which are just, I think those are words that like developmental psychologists have really applied to this selection, this description conversation that we had. Candace is going to talk a lot more about this. I just wanted to show you the other thing that we had, which was just that our kids' genes are predictive of their parents' education levels, their mother and their father's. And luckily right now Fragile Families is getting the mom's DNA. We already have the DNA, but doing all their genotyping too, so we'll have the moms and kids.
Sorry, can you
that
go
gets
back
to
one?
the kids. Yeah.
So evocative. So like if I have DNA that makes me hideously ugly or something,
Yes.
then I
I disagree with what you're saying, but yes, go ahead. I understand.
Well,
It's like the third time.
because I'm trying to.
Anyone who's married to Margot would feel the same.
I'm trying to pick something that would have range within race, but that should not actually be affected, or should not actually be affecting your cognitive performance.
Right.
But it could evoke an environmental response where people don't sit down next to you and maybe tutor you, mentor you, and put you in leadership positions and so on.
Right.
And as a result, you then don't get exposed to curricular opportunities, and then you attain less. Those genes might be picked up in the polygenic score.
Sure.
But the mechanism would be something about evoking a response from the environment.
Versus you actively going and doing, you know, your preference is choosing something else.
And so I feel like it might be helpful when explaining this line of work to new people to think of aspects of the polygenic score that are likely directly related to the domain, or that, given how we structure society and the way in which we allocate educational resources, could be consistently evoking a certain kind of response. But you could imagine a counterfactual world in which that maybe there's no reason why attractiveness should be related to outcomes.
The example that I usually give when I'm trying to describe this is just saying that sex is a genotype, right? And you could use that genotype and predict educational attainment like really well 50 years ago, and that's both shorter than it was now, but it's not because we think that biology has changed, it's just we live in a much different environment, and being a woman in an educational environment today is very different than it was 100 years ago. And so the same sorts of patterns could emerge, but just in maybe more subtle ways.
I want to make sure Candace gets plenty of opportunities. This is the set of reviews, so you guys can see all the slides. This is all mostly candidate gene environment interactions, but a lot of discussions. So someone had mentioned like, it's Alex, like all the different types of models of gene environment interaction. And I think the thing, and I contributed to this, let me just be honest, a fair amount. So, one of the things I felt very strongly after like thinking about all these really complex things is that at the end of the day, it kind of comes back to a basic idea that, and this is interesting considering we're talking about interventions now, but a lot of the gene environment world and interactions in general were built on intuition that came from experiments about these really, polar experimental designs and how you can detect that interaction. The reality is we're often not seeing that in observational data, and so we're not actually often powered to test interactions that we think we're powered to test. And this came up earlier today. And so the rule of thumb is generally, if you see an effect size, a main effect size, if you want to see an interaction of the exact same size, it's about 4 times the sample size you're going to need to detect that. If it's half the size, it's 16 times the size, sample size. So it rapidly gets out of hand, which is why I think, as exciting as it sounds to look for an interaction, I would strongly suggest that we continue to follow what we've been talking about before, which is more about mechanisms and selection. I think we're a ways away from the full interactions. And I think that's Candace.
So I know we're running short on time. I just wanted to kind of make 3 points. I think that came up, or 2 points that came up in our group, and then share a couple of slides from a paper that Dan and I did. And the first one is really just to kind of channel Greg Duncan for a second. This was a slide from their book, and we talked about selection into environments. And I think an important kind of message from the world of context, so Sean Reardon has written about this, Greg and colleagues, that children are increasingly becoming segregated by income and by race in their schools, in their neighborhoods, in the United States for sure. And this shows over time in terms of parental financial investments, right? So if you just follow the bars over, this is 1972, this is average spending per child. You see kind of a flat line among the lower incomes, those are the lower 20%. And then you see just this kind of rapid rise, so how much we're channeling into each one of our children. And so just to kind of put this in the kind of perspective of thinking about modeling the genetic effects, selection effects, right? So the children are becoming increasingly similar in lots of ways across settings and could be amplified in their genetic markers we get across time. I think we've talked a lot about new tools to cheaply gather information on genes, right? And that is also the case for becoming increasingly possible in context, right? So we have new tools to kind of advance our measurement of neighborhoods. So this is from, Dan, I've seen this. This is a map drawn by hand of London streets by poverty levels, right, in the 1800s. Now we have these same kind of procedures that go kind of large-scale mapping from large-scale big data sets, and then increasingly using AI, right, to kind of crawl through publicly available data sets on geography, right? So if you think, kind of to tee up for the conversation about what the future might look like, so what would a future look like in terms of when 23andMe, all the DNA is uploaded with, and everyone sends it in with the address, right? And we can crawl through with machine learning, right? Are the kinds of conversations we've been having for the last 2 days impossible for us to have because we can't do the 50-way interaction that is likely the model of the world, right? Like, I mean, are we waiting for this to figure out some of those mechanism things, and then what can we do in the box that we're in now with the tools that we have? So kind of just setting that up. I wanted to show, these are Dan's slides. So all of the results that he puts on one slide, he also has broken them up into individual slides. So instead of like, is it slide porn? This is like kind of the slow rollout here. And so this is just going to be and so the other thing I want to pause and say that it's actually really weird from an intervention and social policy perspective to talk about a study of neighborhoods that includes only people of Caucasian ancestry. So if we're thinking about, you know, using genetic information to kind of understand neighborhoods and, you know, genes or selection, that's great from a scientific perspective. If we think about every large-scale intervention, including randomized housing vouchers, those have been among low-income African-American population. I mean, so we are worlds apart right here. So just to be clear about, you know, this is, you know, the implications of this for policy are ways off. So I'll just skip to this slide. So this is a British study, you know, in terms of ancestry. And in terms of neighborhood composition. These are Dan's slides, so Dan can correct me if I go wrong here. This is just the classic thing that you always see. These are the wealthy neighborhoods. These are the most deprived. 2 measures of how wealthy your neighborhood is. And then here are all the different outcomes that we looked at. So if we look at obesity, we look at mental health, we look at early childbearing, we look at educational success. You see the gradient, right? So this is the gradient. So in our study too, this gradient exists. On these same individuals, we also have the polygenic risk score. So this is just to assure you that that also predicts for the kind of outcomes that we have here. So this is the relationship between polygenic risk score for obesity, for schizophrenia, age at first birth, educational attainment,
And educational.
with not in education, employment or training, so need status. Anything to add there? Kind of differing levels of kind of predictive power, but you can see that the educational attainment has the strongest one. So this is the main take-home slide finding, and the question really is, when you have a child's genotype, when you genotype them, is there selection into neighborhood context? So are kids that are born with a higher risk for physical health problems, for educational failure, born into poor neighborhoods? And so we have mixed results, right? And so this first slide here is obesity, and the red line is the association with the polygenic risk score. So remember, this is the wealthy neighborhoods, the most poorest neighborhoods, really no association for polygenic risk score, right? This blue line is that relationship that we're finding between obesity itself and neighborhood, right? So we still see a gradient in obesity by neighborhood, we know that, but it's not being, we can't account for that based on the polygenic risk score. If we move over here to mental health problems, we see kind of a stronger relationship. Again, the correlation here is 0.04, so we're not talking about huge effects, but we're seeing, again, here's the polygenic risk score. My eyes are gone, so.
So what's going on here is there are 2 different measures of neighborhood that we use. one of the measures was a kind of off-the-shelf commercial classification of neighborhoods. They're not, I mean, like small areas in the United Kingdom by Acorn, which Candace and Temi put together. And then the second one was kind of a boutique measurement that we built from a lot of data that Candace had accumulated about the neighborhoods where people lived, including police records and her systematic social observation, where her students have done virtual walks through the neighborhood. So we had fairly involved measurement. And so what you're seeing on the two sides of each of these figures are results presented for, on the left side, the Acorn classification, and on the right side, this ecological risk index that we built. And in fact, the initial vision was to define like 5 dimensions of neighborhood risk, but it turned out they were all correlated at like 0.8. So there's like one general factor of like living in a high-risk neighborhood, and that's what's being quantified there. And so you can think of it as being kind of a method check. We should see the same result across both of those panels, and so we should have some concern about the robustness of the any apparent gene-environment correlation with schizophrenia polygenic score, given that we're essentially not seeing it replicated across the two. For those of you who have seen the Sari Eslan paper reporting gene-environment correlation with the schizophrenia polygenic score, the magnitude of the RGE reported in that paper, in fact, is 0.04. It's just that he's analyzing a much larger sample. He reports it in terms of R squared, but basically we're getting the same effect size that he's getting. It's just a question of whether you think it not being 0 is.
What's the phenotypic correlate? I mean, I don't know how to interpret these.
Yeah. So what you're seeing, the blue line is showing you the increase in the fraction of kids who are obese as you move across the distribution of neighborhood risk from low to high.
Oh, okay. So blue is okay.
And the red line is showing you the average polygenic risk for obesity in kids living in neighborhoods from high risk to low risk. So essentially, genetic risk looks the same in poor neighborhoods and rich neighborhoods in this sample. Phenotypic risk does not.
Very helpful.
Thanks.
Sorry, this is a lot we have a lot of trouble.
Okay, so here we have teen pregnancy, so same thing, 2 different measures of neighborhood, and you can see here's the phenotypic correlation, here's the polygenic risk score correlation, right? And now we're getting correlations of up to 0.12, and then these are the educational measures, and here we have 2 blue phenotypic lines. One is for no educational attainment, and then the other one is for this NEAT measure, so not in education, employment, or training. And then here's the polygenic risk score, right? So we're seeing this.
So, okay, these are all whites.
Yes.
Okay, and then the high poverty neighborhoods have a lower proportion of whites.
This is England.
This is England.
Oh, it's England.
Right,
Oh,
and so,
okay.
so in some ways, it's been nice to do the neighborhood work in England, because when you do it in the United States, there's, it's so confounded in terms of race and SES.
Right, because at least consciously people make choices of where to live,
Right.
in part based on the diversity of the neighborhood, positive or negative arguments.
Right.
Like they are for
Right.
the white flight suburban.
Yeah.
Okay, so you're not confounding choosing to live in a segregated neighborhood or willing to live in a non-segregated neighborhood with risk in this, because it's.
Well, it certainly is a story, it's all of those things. I think it's a story about selection, and so we have the mother's polygenic risk score. We see that those are associated, I don't know if these those are also associated with kind of, so this goes across generations, right? And
So what we find is that, I don't know if you have the slide, but if, a question that you might ask is, well, these kids didn't pick these neighborhoods, right? So where does this gene environment correlation come from? Yes, this is a little different. There it is, yeah. So this is the magnitude of the effect when you condition on the mother's score. So the light blue line is the, you get rid of most of the RGE when you account for the mother's polygenic score. Presumably if we had the dad's, we'd get rid of all. Because it's possible that there is some selection of families into neighborhoods that are near services for kids who need services. So one of the hypotheses for why people with schizophrenia tend to live in urban areas is that they move to places where there are psychiatric hospitals because that's where they're getting treatment. That could happen with kids who have various kinds of health problems, but I mean, it looks like what we would expect is that.
Can I make
just related to this, just going back to when we were talking about applying for funding and writing proposals and stuff like this, I think that this highlights, again, when we're talking about genotype, like we should genotype our samples, we should also genotype our parents if we can, right? Just given that these big biases exist, because we should probably, try to incorporate both if you can.
So I think with looking at the clock, I'm gonna stop there. I think that kind of gives you a flavor for kind of this disconnect between where the science and the neighborhood research is and where the measures would need to be to actually say anything that's really policy relevant around equality.
You should show them the bar graph though, the other bar graph, this
Right.
one.
Yeah.
So it's not all genetic. Genetics explain only a fraction of the neighborhood.
Yeah. Right, so one of the concerns is that we would go in and find all the effects are selection, right? That there's no effective neighborhood. And so when we look at the physical health outcomes, it's reassuring because we have this gradient around neighborhoods and we're intervening on the neighborhood effect. We think that that's not due to the selection we can measure in this way, so it provides some assurance, although with the next form, who knows? But here you're seeing that, it's certainly not accounting for the entire gradient that we're seeing.
Yeah, I mean, I think it is worth noting that the prediction R squared for the precision of the BMI polygenic score for predicting BMI, particularly in young people, is comparable to the precision of the educational attainment polygenic score for predicting educational attainment. So these results aren't an artifact of using weak polygenic scores for health and strong polygenic scores for social outcomes. It's really that people sort into neighborhoods on the basis of their socioeconomic attainment, not on the basis of their BMI.
Right.
You don't have like a way in to get into your neighborhood, but you have a buy-in.
And so, is it plausible to say that these, if anything, are under like a lower bar of the true correlation because both things are measured possibly with error, and so the correlation with two measure? Okay.
Yeah, we might expect these two.
Right.
To do a back of the envelope calculation for how much of the neighborhood, to bound plausibly how much of the neighborhood is.
It's in the paper.
Oh,
Off the top of my head.
I swear I read.
I think it's in the supplement, but it might also be in the discussion. We essentially say this is how big this would have to be. I'll pull it.
No, that's right. So one is an observation that it seems like this is another use of genetic data that's similar to what Alex was saying, which is like if I want to study parenting, then the twin design helps me understand this other part. Here too, it seems like if you want to understand neighborhoods, if your research question is only about neighborhoods even, this is very useful. So that feels like a section of educating funders, so that it's just another case of that from mature.
Yeah, I think, I worry a little, just this gets into the broader kind of issue, the politics of interventions around around place, right, are fraught. And thinking about what's the added value of the genetics here, above what we could measure with other things, right? And if that would be the most compelling argument that I would take in.
I think partly what we're aiming to do here is make a rhetorical point about the presence of selection, a way of quantifying selection, and in an effort to recenter some of the conversation in social science, around nature-nurture issues, where there are regions of the social sciences in which nature is sort of not considered as a factor, and I think one of the things that's being illustrated in these data is that it's part of the story. It's not to be ignored. You can't simply put up a correlation between neighborhood risk and some phenotype and assert that this is a causal neighborhood effect. And responsible social scientists aren't doing this, and at the same time, the New York Times is publishing maps that are suggesting this, and in fact, policymakers are acting on those maps to allocate public housing resources.
You guys are preaching to the choir, but one thing Chetty does is he looks at louvers. Can you do that?
So now we're in the world of America. So we do have concerns about how race is affecting these rules. But that's it. So this is at health. These are kids in which we're quantifying their baseline neighborhood risk as the neighborhood where they were living when they enrolled in the study. So they were in secondary school living with their parents, and we have census data to describe essentially the level of poverty in the census tract where they were sampled from. And then at wave 4, we have their address where they were surveyed when they are in their 30s. And we have the same census tract measures, this time coming from a 5-year ACS data set. So the American Community Survey from, I don't know, like 2005 to 10 or something like that.
Right.
And so what we're showing you here is the same thing I showed you in the social mobility plot, but didn't describe, is that the box plots on the bottom are illustrating this very subtle gene environment correlation present at baseline, and that kids living in higher risk neighborhoods tend to have slightly higher genetic risk for early age of first birth, slightly higher genetic risk for poor educational outcomes. But that if we look at them across development, following them from secondary school into young adulthood, kids with higher educational attainment polygenic scores tend to move to lower risk neighborhoods. Kids with higher or lower age at first birth, higher age at first birth polygenic scores tend to move to lower risk neighborhoods. So, children's genetics are influencing or associated with the kinds of neighborhoods they choose to live in when they leave their parents' homes or are forced to live in, I suppose.
What about, so what Chetty does is he looks at people who are moving their kids.
Right, so and the power of his stuff is also within family, right? So there's 2 pieces of evidence, right, that are compelling, and there's like the mover stayer model, which is a classic kind of way to look at this, which we don't have, enough movement in the early years to do that. But then it's a beautiful example for him. He goes back and does the moving to opportunity study, and he looks at effects within family, which is kind of what we've been talking about here, is the power of isolating that within family kind of effect, because you are able to hold constant by design for the rest of it.
Could you concentrate the ad health thing just in the states that are predominantly white? I mean, we are the most diverse country in the world, but we're still almost 70% white, and that diversity is not randomly distributed across the country. I mean, there's many states, as we know from recent elections, that are overwhelmingly white. And if you think about, because this idea that people are geographically mobile is something of a myth.
Yeah.
You going from North Carolina to California makes you the exception to the exception to the exception.
Yeah.
And so most of the movement, like what Chetty was doing, is really just in small bounds. And if you think about the region that you're in or the state you're in as fairly exogenous, much more so than your neighborhood. And then within that, it takes out some level of.
Right.
So
fixed effect.
We can do this
that's related to race.
We can do this with state fixed effects.
Yeah.
I don't think we actually do that in the paper. It doesn't change the results. I think we've even played around with doing it with school fixed effects.
Yeah.
Although there aren't that many schools that are sampling from enough tracts to get that much variation.
Yeah.
But looking at
struggling
states,
with whether fixed effects is
right. You just want to concentrate on
yeah.
Midwestern states, upper Midwestern states.
Yeah.
And then it's just a question of where are those ad health saturation schools? So it's a school-based sampling frame in ad health in which there are a handful of schools that account for a big chunk of the participants. And so if they happen to be in especially white states, then they should be in business. But if they're not, I would not be surprised if they were not. Then focusing on those states may limit the amount of variation we're going to have, across tracts.
Why do you need the saturated? Well, it's
just a sample size. As it is. Yeah. If you pull kids out of.
Actually, that's not entirely true. There are a bunch of small schools that weren't designed to be saturated, but by default because of we use them in the friendship paper. And I think there are about 10 of them, and they are disproportionately white.